Everybody, this is the walkthrough video for Wednesday. It's November 12th, and I'm looking for you to find your yellow bell work. Here is the picture that you should kind of have in your head. You don't need to draw it on the bell work for today. Here, I'm looking for a review from you of which things bind to which and how many hydrogen bonds they use. Just a reminder that you can use A and T and C and G in your answer. So go ahead and pause the video and take care of that. All right, so you can write SAME, S-A-M-E, as yesterday. That's going to help us remember that this particular learning goal we're carrying over these 2 days. So today we're going to finish medical mutations case studies, and then we're going to begin the final report for medical mutations. Students who are working on time are going to finish today. Students who might need a little extra time, I'll give you in-class time tomorrow. If you're somebody who's really working behind, you are probably going to need to work a little harder outside of school just to make sure that you're finishing things. This class is made so everybody can be successful, but people work and learn at different rates, and you might need to put in some time outside of class. Excuse me. Let's go ahead and check our colorful table of contents page. Today, I think it's pink. Not today, like it's always pink. This trimester, I think it's pink. And in a moment, it's going to go ahead and load. It's going to be really glorious when that happens. Here we go. So I'm going to scroll down here and show you today's paper is page 10, final report, medical mutations. You can write this out on a piece of binder paper and just staple it. You can look online and see what it asks you to do, and then just fill those out and staple it to the back of an existing packet. So I have the questions and your work together, because some of the assignments, this one has text in it that's important about case studies, for example, or this one has text in it about enzyme shape. So, all right, cool. Ooh, bird clock. Okay, so where have we been? We learned that enzymes do important jobs, and we thought about what would happen if our enzyme had been a different shape. And we remembered that a protein's shape is what makes its job possible, right? If you change the shape, like with pH, you don't really have an enzyme that can do the same thing, right? And so we had this big question: how do proteins get the shapes they need to do their jobs in living things? And we realized it's all in the DNA. So you learned about genes, which are the sequences that encode for proteins. You learned they had a start codon and a stop codon. You learned that they are on the DNA, on chromosomes. You learned that all living things have DNA. And you explored the structure and base pairing of DNA in this assignment, right? And then we said, okay, well, if DNA is important, how do you get the information from DNA in order to make a protein? And we learned about that. Transcription is the process where the DNA information becomes messenger RNA, and translation is the part where messenger RNA becomes the information in proteins. This is a ribosome. It's a little machine in the middle. Oh, the door is running. It's a little machine. It's throughout all of your cells, and it's in the process of making protein. Cool. School is haunted, it's fine. So you learned about transcription and translation. Those ribosomes are the factories where proteins are made. If you take AP Bio with me in your junior or senior year and do college level bio, you'll learn more about that. So we talked a lot about how if there's DNA and there's a typical gene, you make a typical protein, it does a typical job. But changes in DNA, mutations, can cause a mutant protein. Now, sometimes that mutant protein works even better, but a lot of the time, that change can cause a disease phenotype or a medical condition. And so we've been learning what happens to people when this happens. So first things first, we're going to check the answers to this assignment: DNA to messenger RNA to protein, transcription and translation. And I wanted to say to you, we're going to talk about the answers here. I'm going to use that annotation tool because I'm learning how to do that, excuse me, like a boss. And I wanted to say to you, I'm only looking for the simulation answers and these answers, but you should be able to know how to make, if you're given a strand of DNA, to make the complementary strand where A binds to T and T binds to A. You should be able to take that strand and make a messenger RNA strand where if you see an A, you put a U. If you see a C, you put a G. And then you should be able to use a codon table of your choice, either the wheelie one or the square one. And you should be able to read off the codons, those things. Now, I want to remind you, it's up on the board behind me. Maybe we will take a little stroll here. Hang on a second. Maybe we will take a little stroll here just as soon as I remember how to pause sharing. Great. So now I think you just see my face. I hope you just see my face. Hang on a second while I figure that out. What a face, too. Am I right? Yeah, I think we're, wait, what's it showing you? Great. Is it just me? I think it's just me. Okay, great. So I'm gonna take you over here, little field trip. Let's see if I can move y'all. Do you see what's written there? See how it says 1 codon equals 3 nucleotides equals 1 amino acid? I wrote that down on the board today because it is going to be important for us that you know that information. So back to my there. Back to my annotations. How many codons equal 1 amino acid? Well, codon is the triplet code of letters of nitrogenous bases. So the answer here is 1 codon equals 1 amino acid. A protein is a sequence of polypeptides is when there are lots of pieces of one protein, like lots of strands of one protein. But a protein by itself is a sequence of amino acids are those building blocks. Ribosomes are the place where which takes place. A lot of people remember that it's translation. So you have to memorize that part. How many bases equal 1 amino acid? Well, it's 1 codon, but 1 codon is 3 DNA bases that equal 1 amino acid. Sometimes people think that because this is true, that there are 3 different amino acids in humans, but that's incorrect. There are 20 different amino acids. They're all the ones that are on that codon table. What's it called when there's a change in the DNA code? That is a mutation. Okay, and I'm just going to tell you these so you have these facts in your notebook. Remember, your notebook or your binder is like how we're making the textbook. When there's a mutation in DNA, the change is permanent, and all of the proteins made afterwards will be affected. And also, when there's a mutation in the DNA, only one gene is affected. All the other genes are still there. Sometimes how you turn genes on and off has changed, but the important part here is a mutation affects only one protein. And that's important because as you're learning from your case studies, one protein can make really serious effects as a result. So just a reminder, I'm only looking for these answers for a full credit paper in addition to like where you filled in the blanks during the simulation. So I'm not checking the transcription and translation. Please make sure that you are able to do that and you know to stop when you are given a codon that has a stop in it. Okay, cool. I have to learn to clear all the things. Great, look, I'm learning. So proud of me. All right, next up, we're gonna talk together about, Gary, next up, we're gonna talk together about the case studies assignment. So remember, case studies are like detective stories for doctors and medical researchers. They help those specialists learn about real people and the medical problems they face. So all of you were investigating a case study about one medical patient with a condition that is caused by a change in their DNA. I know it says mutation in their DNA. So, you know, these I can statements I'm giving you, they're coming from the state of Indiana, and they're helping us think about how the work you do in class is tied to what the state asks you to be able to do at the end of a biology course in high school. In this case, we're identifying a mutation in a DNA sequence. We're describing how a mutation can alter protein structure and function. And we're explaining how a change in protein structure can lead to a disease. So where are we going? You're going to do the assignment called Final Report Medical Mutations. It's going to have you write down the patient name. I think it's Stuart, Kim and Aaron, Kendrick, and Chad, right? So you're going to pick the person that you researched, and then you're going to write your last name. I'm Dr. Milks. I'm not a medical doctor. I have a training in biochemistry, but you're going to pretend that you're a doctor. You've grown up, you've done college, you've gone to med school. Here you are writing for a journal like the Journal of the American Medical Association or the Lancet. And so I don't know if you'll notice, this assignment is just rewriting answers that you already wrote down on yesterday's assignment. If you haven't finished that one, you have to finish it first in your work time for today. Now, the final report medical mutations may look difficult, but if you've been working hard and staying focused, you already have all the answers you need. There's another really important thing. It says that people can type. That is if you have an IEP or a 504 this year. We noticed that people were just going online, asking chat and writing the answers down. Chat's going to give you information like the Snapchat, ChatGPT models or the AI character models. They're going to, or Gemini or whoever you use, they're going to give you information that's above and beyond what we're learning in this course. And I'll actually ask you to redo it using specifically data from the case study cards that you've been given. So before you get to that, I wanted to remind you, you wrote down a summary here about the medical symptoms, about the DNA changes and about what happens in proteins and or cells, this paper is going to ask you those same questions, right? You've already written this even in this little organizer. You wrote that up in your assignment. So hopefully by now you're pretty practiced and you've had enough practice that you can turn it into sentences. Now there's not a lot of room in the worksheet. My chair is moving down. There's not a lot of room in the worksheet, and that's on purpose because the goal here is to have you practice summarizing really succinctly and really clearly. So I want to show you a happy picture. This is a picture of Kendrick. So if you were one of the people that studied Kendrick, you know that his childhood, he was just in intense pain all the time. He did not get to do the things that kids usually get to do, like riding bicycles or playing basketball. His family is in the back. Everyone is super joyous because this is a picture taken of him about a year ago. Life Without Sickle Cell, Beckon's boy who completed gene therapy. So if you're interested, clicking on the text here and the slides, which you can reach in today's date link, will take you to a gift link. So you don't need access to this. You'll be able to see it. So after 44 days, Kendrick left the hospital. While his family feels fortunate he was the first to receive a treatment, their difficult experiences hint at what others will be up against. So they're very celebratory of Kendrick here. This is what Kendrick looks like after his treatment. He's your age now. He's like 14 or 15 now. And those of you who learned about it know that he had a really difficult childhood, searing pain, and the only way to fix it was to be hospitalized. So let me tell you about sickle cell. About 100,000 people in the US have sickle cell disease. And for the 20,000 or so with the most severe disease, gene therapy, advances in medicine, may be their only hope of living a normal life. So you learned that the disease, sickle cell, is caused by a mutation in hemoglobin genes, if you research this one, that leads to crescent-shaped red blood cells, like croissants, which tend to get stuck in blood vessels, causing episodes of excruciating pain. And the blockages can damage organs, they can cause strokes, and shorten lives. So until recently, that's what life was like if you ended up with this mutation twice in your genome. And then last year, it turns out that people have been treated, and Kendrick was the first one. So what's the treatment look like? So he went to the hospital. His doctors removed stem cells from his bone marrow, and they sent them to New Jersey, where technicians added new healthy hemoglobin genes, and then the cells come back to the hospital. And then they killed every single cell in his bone marrow, and they put the new ones back in. He was very, very sick. He was very, very sick. It was not an easy time for him. He couldn't eat. He couldn't talk. He was tortured by the pain, and his parents were too. And it was successful. So when we think about what the potential is for medicine in the world, I want to be clear, medicine can help people. And in fact, one part of today's assignment is going to ask you to look back over the case study. They're all linked in Tuesday's Canvas. And you'll be able to think about what medical innovations or like patient support is helping people live their best lives when they have these mutations. So at the end of this assignment, you should know how to do these things. And then here's the cool down for today: the name of the condition you ended up researching and the symptoms of those conditions. And if you're just joining us, I should say this to you, you probably want to focus on Kendrick. His is the most straightforward of the situations. All right, that's it for me. I hope you're doing well out there. I'll see you soon. Bye.